share. Give me a second. I hope everybody can see. Am I sharing?
I can't see anything yet, Marija.
Not yet.
Okay, let's try that again.
Yes, we can see your screen.
Yeah, and the presentation mode, good. All right, I can see now.
Yes.
All right. Thank you, Colette, for that introduction. I'm going to switch off the camera so that we have a better connection. You can hear me better. Hope everybody is nice and warm and everybody has a nice hot cup of tea or coffee or chocolate so that you can enjoy and relax. Thank you everybody for joining us. Like Colette mentioned, we are doing the NGS series again. We are back by popular demand. We've done this last year and we had a huge success and we want to continue where we left off. So we're gonna, for the beginning, we're gonna start talking about infectious diseases, but please join us in other sessions as well, where we're going to talk about RNA sequencing, a little bit of bioinformatics, but also talk about NextSeq 2000 and the NovaSeq. So please join us for other sessions as well. Thank you everybody for joining, and let's start. Like Colette said, my name is Marija Kvas, and I'm Senior Application Specialist here at Separations. Let me just get my pointer. Good, all good to go. So when we talk about NGS and Illumina, the main goal of Illumina is to improve human health by unlocking the power of the genome. And we can do that directly by looking in human health, right? So looking into, for example, reproductive health, where we're talking about non-invasive prenatal testing of the expecting moms, or talking about looking at different cancer markers when we're talking to oncology patients, or we can do it indirectly looking into agriculturally important species and making sure that our food is up to the standard, improving our food, through market-assisted breeding and so on. But today we're going to focus on infectious diseases. We will touch on infectious diseases, but techniques and applications that we talk about are obviously applicable to any microbes, small genomes out there that you guys are working with. So why is Illumina such a important, having such important technology, and why is it so widespread globally? Over 90%, more than 90% of NGS out there happens on Illumina platforms. And this is because we have an integrated sample to answer workflow. We have a vast amount of different library preps that allows us to create libraries, so to make sure that the DNA is sequence ready. And then obviously we have a portfolio of different instruments that we can use to sequence this data, and we have analysis pipelines such as base cloud server, also a Dragon analysis pipeline, which is a ultra fast processing analysis pipelines out there that we can utilize to do it. So we have this from the beginning to the end solution, where we're talking about simplified library prep. We can also do a certain custom contact, but also we're trying to make it as economically viable as possible, and to have the full solution with integrated analysis. So why is such a big boom when when it comes to using NGS in a different labs? Obviously, we can sequence so much more, right? We've been sequencing for the past 40 years or so. It's not new to us, but the way we do it with NGS, it's this big boom of data that we can get. So it's a large amounts of data that we can do. We can simultaneously do multiple different samples, multiple different organisms, targets. So it allows us to massively parallel sequence many different things, but at the same time to manage that price per sample cost and to reserve the precious samples that we have. But we have incredible power of sensitivity and data resolution when it comes to NGS, because we can sequence so much more, so much deeper. It allows us to have that extra confidence when it comes to our data analysis. So to compare it to the other well-known technologies out there, like most of you know, PCR, qPCR, Sanger sequencing, Obviously, NGS, because we can do so much, we can sequence so much, we can do a whole genome at the same, all at once. We can do multiple samples all at once. We have that extended discovery power, but we still maintain that resolution and high sensitivity, something that we cannot do with qPCR and Sanger, right? With those two techniques, we have low discovery power. We can only focus on a couple of targets at the same time. And, if we are increasing the number of targets that we deal with, obviously that increases the cost, but we also have a low variant resolution. We have a low variant resolution, and we have lower sensitivity when it comes to these technologies, right? So with NGS, because we don't have to have any prior knowledge of what we are sequencing, right? We don't have to have any idea of what is in our sample. We are able to discover novel organisms, novel pathogens, right? But we're still highly sensitive, because at the same time, while we are looking at the specific pathogen, we can also look into co-infections, quasi-species, and we have that high resolution of data for genomic comparisons, mutation detections, and characterization. And like I said, we can put simultaneously, we can sequence more than one sample, more than one organism on one run, and that means that we're sharing that cost of that sequencing kit, right? With qPCR and Sanger, every time we want to do additional target, additional gene region, it's an extra cost, right? It multiplies the cost. But with the NGS, more samples we have, more targets we have, obviously we are sharing the cost, and it becomes actually more cost effective per sample to do it, provided that we have output of data that we need for each sample. And obviously this then, because we can sequence so much deeper and wider, this allows us then to also study pathogen host interactions at such a much deeper level. It comes to, again, looking at NGS versus this other traditional methods, not just molecular methods, but also culturing out there and analysis. Some of those methods are obviously limited when it comes to species identification, and also to do it simultaneously would require more time, more resources, right? But because we can do more at the same time with NGS, this allows us to look at microbial community as a whole and understand interactions between them, and also to identify microbial at a lower level, right? So something that is not that abundant in a sample, because we are sequencing deeper, we can still get to that specific individual and pick it up, right? So that's where that high resolution comes in, and that high sensitivity that we have with NGS. I just briefly wanted to show you a different portfolio of instruments that we have out there. We have benchtop instruments that are ideal for what we call a small genome sequencing. So your infectious diseases or any microbes out there. That doesn't have to be infectious. There's also a good microbes out there. But these iSeq, MiniSeq, MySeq, NextSeq are ideal for those genomes, right? They are smaller capacity, and instruments, you will notice that iSeq produces only 1.2 gigs of data. MySeq, that is a very common and I think most popular instrument out there globally, but also in South Africa, it can go up to 15 billion bases that we can generate on it. And then we have larger instruments with which we can do so much more, right? Not only microbial sequencing or extreme number of samples that we can do on it, but also for the larger, agricultural important species, or even for human health, right? A very popular is human whole exome sequencing, whole genome sequencing, and so on. But we'll touch on those applications in the later series. Today we will focus on the small microbes. So when it comes to applying genomics in infectious diseases, obviously the first thing would be to identify what is the organism that we are working with, what is that pathogen. We need to identify it, right? And we need to characterize it and understand it, right? We need to understand its evolution and how is it developing in order for us to to understand where it obviously came from, but also to track that transmission, to monitor, to understand the epidemiology of that disease and pathogen, but also to understand the virulence and pathogenicity, and how does that affect host immunity, either human or animal, or how does it affect maybe even genetics, and that will help us. Obviously, the final goal would be to develop prophylaxis, to develop treatments and vaccines against this infectious disease. We're also looking into animal health. It's really important that we understand what are veterinary important diseases out there. How do we manage those outbreaks and make sure, that we don't have a zoonotic jump? But also, it's really important that we understand that food, what pathogens we can find in our food, because obviously that has the direct impact on our food safety and security. So it's really important that we monitor animal health, but also food production and food quality. And when it comes to environmental surveillance, of course, we need to understand which animals out there are potential reservoirs of different infectious diseases, and what is the potential transmission vectors that we have out there. Obviously, we can also monitor antimicrobial resistance and what genes are out there available, present in the different environments, either being soil or wastewater, for example, something that is really popular looking into. So we need to understand our microbes in order for us to develop strategies to obviously monitor and control them and contain them, right? So when it comes to NGS in microbiology, we're talking about obviously identifying those microbes, discovering them, right, and characterizing them, understanding what genes they have, what, maybe antimicrobial resistance genes they have, what is the virulence, pathogenicity, maybe even toxicity to humans, and at the same time we want to understand mutation of that pathogen, right? We need to identify different variants, mutants. So we need to understand how is it evolving through time, and what are the rearrangements that we are seeing, and obviously then how does it infect the host, either being human or let's say agriculturally important animal, right? So we need to understand what is that human pathogen interaction that is happening, which will then help us to monitor that disease, to control it, to ideally prevent it, right? Because ideally we want to prevent any disease out there. So when it comes to applying genomics in infectious diseases, of course, like we said, we need to identify and characterize. We need to understand the mechanisms of that transmission and understand how is that pathogen being transmitted between organisms. And we need to monitor for different variants, but at the same time, we need to understand the zoonotic reservoirs that are out there, and what animals are those reservoirs, what are the vectors that we are dealing with, so that we can monitor the spread of this disease, jumping between reservoirs or hosts or carriers, right? And all that because obviously we want to develop a therapeutics, we want to develop a new treatment, new vaccine, antimicrobials, even maybe probiotics. So it's really important for us to understand, monitor and control the infectious diseases, because we need to prepare for a new emerging pathogens, right? But also to look at the transmission risk and to have a quick response to the future outbreaks, right? And for us to do that, obviously, we need to monitor those pathogens, understand the movement of those pathogens through movement of animals, and to understand the pathogen evolution, and also to control the vectors, right? Those vectors that are transmitting, let's say, the virus between animal reservoir, animal host and a human host, for example. And at the same time, we need to educate the public, right? And we need to limit that spread of the infectious diseases, limit the human contact. And obviously, we want to have a quick response to future outbreaks out there, not just like we are dealing now with SARS-CoV-2, but other, let's say, foodborne pathogens. Obviously, we want to stop that spread, and we want to timely react quickly in order to stop that spread of infection, and also to have a fast development of some kind of prophylaxis, right? Either being a treatment drug or ideally a vaccine. So when it comes to methods out there, when we're working with microbes in microbiology, infectious diseases, or industrial important microbes, for example, I would say that foremost core methods that we will touch on just now and we will deal with in a little bit more detail would be obviously whole genome sequencing. That's the ideal, right? If everybody could do whole genome sequencing, that would be great. It means that obviously we are sequencing all the bases of that specific organism, so the whole genome, either something that we are familiar with and we are just, let's say, resequencing and mapping it against the reference, or we are doing it completely new, describing a new species, doing it de novo, right? Then we have a shotgun metagenomics approach, which is basically a whole genome sequencing, but of environmental samples, right? We are dealing with, let's say, wastewater, soil, let's say, gut microbiome. So we have a mixed population of different organisms in that sample, right? And we are sequencing everything that is in that specific sample and all the genes. So we're doing a whole genome of that specific set of microbes that is in that specific sample, right? Then we have what we call, obviously, amplicon sequencing. This we are very familiar with. You guys are familiar with barcoding. We've done this for years with Sanger sequencing, where we focused on 16S rRNA metagenomics for bacteria, or ITS region for fungi. So we are focusing on specific type of organisms that we want to sequence, right? So it means we need much less data, and we are focusing to a specific group of organisms that we are interested in. And then we have a method, enrichment method, where we are designing probes, and we are actually capturing, with those probes, we are capturing the regions that we are interested in. So we have a specific target that we are looking into. It can be specific genes that we are interested in, or it can be that we are looking at many different pathogens at the same time. Maybe we are looking into different co-infections and we want to pick up on those. So we'll touch on these in a little bit more detail, right? When it comes to what we mentioned now, whole genome sequencing, amplicon sequencing, and shotgun metagenomics, obviously we're talking about culture independent sequencing versus having a culture, so having a pure organism, right, working with a single individual. Or when it comes to whole genome sequencing, obviously we're looking into food outbreaks and identify those sources, where did that come from, but also monitor the pathogens in the production of food and food facilities, and differentiate what is the persistent pathogens in there, but also monitor those virulent factors and antimicrobial genes, right? When it comes to more targeted approach, like amplicon sequencing, or looking at the variety of the community of different pathogens in there, we can employ shotgun metagenomic sequencing, and there we are looking into community profiling, either just one type of organism, let's say just like bacteria or just fungi, or we are looking into everything that is in that sample, but we are looking at community profiling to predict the pathogens, the spoilage risk, to maybe detect GMOs, to verify speciation. So, for example, in halal food, to make sure that there is no material that is of pork origin, right? So we can employ NGS to do this for us, right? And like I mentioned, we're talking about, either we do it more targeted approach, in which case we are zooming in on a very specific taxa that we want to work in, either bacteria or fungi, or so 16S for bacteria, ITS for fungi, or cytochrome oxidase 1, where we're talking about different animals, maybe protists, right? But we are focusing on a specific, so we are zooming in, we're just looking at the specific community, right? Specific bacterial community, for example, right? With shotgun metagenomics approach, like I said, we are sequencing everything, right? So everything that is in that sample, it can be bacteria, fungi, viruses, certain proteins. Maybe if you're working with soil samples, it can be some nematodes, plant materials. So everything that is in that sample, we will sequence, right? And we require enormous amount of data to do this, right? Because we want to sequence, let's say that you have 10 different species in there. Obviously, we need to now do a whole genome sequencing on all of those 10, right? So it takes incredible amount of data that we need to generate. And obviously, then we need to analyze that incredible amount of data, right? And that is where the fun part starts, right? This is the most intimidating part of NGS is the analysis portion of it, right? And when we are working with so many different organisms at the same time, it becomes more complex to do it, right? Same time, obviously, there's advantages to doing shotgun metagenomics. Obviously, it's culture-free method, right? And we don't have to have any prior knowledge of what we are sequencing. So it's completely hypothesis-free testing. We are simply exploring what is out there. And because we don't have to have any prior knowledge of what is in there, it's ideal for pathogen detection, right? We can discover novel species, right? And this is how, this is what, you know, in December 2019, this is how SARS-CoV-2 was discovered. They did a shotgun metagenomic on a clinical sample, sequenced everything that was in that sample, you know, the nasal swab. And obviously they got some human DNA, there's some bacteria, some viruses, but that's how they discovered a novel SARS-CoV-2 species, right? And also shotgun metagenomics helps us to understand that relative abundance of microbial taxa that is out there, right? And because we are sequencing whole genome, we don't just identify who is in that community, right? Like we would do with more targeted approach. But because we're having a whole genome now, we obviously we understand the whole genetic makeup of those organisms in there, and then we can mine that data to infer potential, let's say, enzymatic functions, or we can look into antimicrobial and virulence factors, right? So it tells us more about the organisms that are out there, not just you know, the names of those organisms, right? So I've been talking a lot about this whole genome sequencing. Of course, it's a I would say a golden standard. If we can do it, everybody would do it. And it's not that we technologically can't do it. Obviously, there's constraints when it comes to a budget. Obviously, if we are working with a small genomes, we are very lucky, right? We can do 100x, 1000x very, very easily. But when we are talking, for example, about human genome, which is 3.3 billion bases, it becomes slightly more costly to do it, right? So then we would look into something like more targeted approach. But when we're talking about whole genome sequencing, obviously we want to map novel genomes out there, novel pathogens, novel organisms, and we want to do comparative study, right? We want to understand what is the differences between these organisms, right? We want to characterize them, look into the identify the mutations that we have, right? And also it allows us to do a low frequency variant exploring and to look at, like I mentioned, that genome rearrangement. And because NGS allows us to do simultaneous sequencing of multiple species, we can obviously do multiple samples at the same time, right? And again, like I said, share that cost of sequencing kits, reagents amongst many different projects. When it comes to how do we prepare our DNA, our little organisms to actually make it sequencing ready? Like I mentioned, Illumina has a vast amount of different library preps that prepare the DNA to be sequence ready. And when it comes to whole genome sequencing, we have something called Illumina DNA Prep, which is an enzymatic based approach to fragmenting the DNA, and to also what we call adding the adapters to our DNA. And those adapters, for those of you that joined us in the previous webinar sessions. We went a little bit into more detail about what the adapters are, but very briefly, adapters allows us to actually bind to the flow cell for DNA actually to be able to be sequenced through Illumina technology, right? So you will notice that we have a little enzyme here bound to the actual bead. So this is how the principle of Illumina DNA prep works, is that we have what we call a transpose. So it's a tagmentase enzyme that will cut, so it will fragment the DNA, and it will also insert what we call a little bag or overhang, and this will serve as a landing site to our adapters, right? So the way we prepare libraries, there's many different steps in preparing the library, but the main goal of the library prep is to have the adapter on our DNA, and that will allow us to sequence it, right? And this specific library prep, the way it's designed, it allows us to have a very versatile DNA RNA input range. Obviously, we can work with RNA, but we need to convert it first to cDNA, right? And then we can work with this library prep, for example, with the RNA, right? But also we can actually, if we are working with a human samples, we can go directly from blood, saliva, or dried spots. We can actually skip the DNA extraction and go directly into library prep. Or if you're working with a pure culture, with a pure bacterial colony, for example, you could also do it, right? But what is main point of this Illumina DNA library prep is that the way it is designed, we have a consistent insert size, so the size of our library is always consistent, and that's important, obviously, when we do sequencing, right? Even, with Illumina sequencing, we're still doing short reads. So it's really important that we kind of match that size of the DNA that we are sequencing versus the length of the reads that we can do on Illumina sequencing. And the way that this library prepares our DNA for that is that it gives it a very consistent size and very consistent uniform size of the library prep, which then obviously gives a very high uniformity of coverage, which then gives it's much easier to assemble if you have a consistent read length, consistent size of the library, and that uniformity. When it comes to de novo sequencing, we have something called Illumina PCR-free library prep. And why this specific, why would we engage into PCR-free library prep? Obviously, PCR has, those polymerases have a specific bias, right? Some regions, they will not amplify as good as they would amplify other regions. And you will notice here on the top in the green, two different library preps that have PCR steps in them. You will notice the white region is where we're having gaps in our assembly, and this is due to the polymerase slippage, right? Because of that PCR bias, right? But if we don't have a PCR step, you will notice that that we don't have any major gaps in our assembly, and that it's more uniform. It's because we don't have that polymerase slippage, and we have the increased indel accuracy, right? And you will notice that we actually decrease the gaps in the final data. So, and this obviously helps with the de novo assembly, because you don't have the reference out there for de novo doing de novo sequencing, right? It's the first time that you are doing this organism. Nobody described this organism. Nobody did a whole genome sequencing of this organism before. So obviously you want to get to the accurate assembly genome of that organism as much as possible, and obviously you don't want to have any gaps, right? And so this is where PCR-free library prep has a huge advantage. When it comes to targeted sequencing, obviously, we want to zoom in, like you saw. We want to zoom in either identifying specific taxa that we want to work with, or we want to look into, let's say, antimicrobial resistance, right? Or any other gene of interest that you know that is important for your research that you are doing. Like I said, very famous out there is 16S rRNA metagenomics sequencing. Like I mentioned, this is nothing new to us. We've done it with Sanger sequencing before. Specifically with Illumina, we are focusing on V3, V4 region, right? So the way it works, we would obviously amplify that V3, V4 region first, and then we would add the adapters to that in order to make it sequencing ready, specifically on a MiSeq, right? When we are working with V3, V4 region of 16S rRNA region, we're talking about 400, 500 base pairs, and that is ideal to do on a MiSeq, which has the longest read length out there on Illumina sequences, right? We can do up to 600 base pairs on the MiSeq, and like I mentioned, obviously you would have environmental sample, right, where you would extract DNA from you know everything that is out there, but you will only amplify a specific region, so 16S only that is found in bacteria and prokaryotes, and you will amplify V3, V4 region, you would prepare the library, you would sequence it, and then we will do the data analysis, and Illumina has a couple of pipelines out there on the base cloud server. So I'm not going to touch on this now, but on the 27th of August, we're going to have a specific session where we're going to talk about the analysis, and we're going to touch on shotgun metagenomics and 16S, for example, metagenomics. So please come and join us then, where we're going to focus more on analysis of data and what is available on the base space. That same approach we can apply for ITS sequencing when it comes to fungi, or any other actual target that you want to amplify and sequence, right? Because we know what that overhang is, right? So we would know the sequence of it, and we would synthesize our primer together with that overhang, right? And the purpose of this overhang is then for the adapters to bind to, right? So when you are PCRing the specific target, the region that you're interested in, you're incorporating in that first PCR, you are incorporating, so you would have a first PCR where you would amplify the specific region, and you would incorporate those tags to your amplicon, and in the subsequent second PCR, you would then have the adapters binding to that overhang to your amplicon, and you would have your library, right? And I mentioned ITS for fungus species, but obviously there are papers out there that are also looking at a large subunit, or they are also looking at the whole, a whole region, right? If you're doing something more than, let's say, 500 base pairs, if we are looking into long range PCR, right? Anything from, let's say, like I said, more than 600 base pairs, but also maybe looking at 10 kb, 30 kb fragments, right? You can also amplify those and then employ Illumina DNA library prep to fragment it and create a library, right? So we can work with amplicons, short ones, or long range PCR products. We just need to know how do we need to modify them in order to be Illumina sequencer. When we're talking about targeted approach, especially in metagenomics, right, when we are working with environmental samples, obviously there are limits to it, right? When we design that primer, it's specific, like I mentioned, it's specific for that group of organisms, either being bacteria, let's say protozoa or nematodes, or whatever that specific primer is looking at. Or if, I'm digressing now, but if we are looking at, let's say, human health, and we are looking at the different genes that are associated with a certain trend, right? Then you can also have that specific primer design to capture those, right? But because we are working with PCR, when it comes, sorry, let's go back to metagenomics and environmental samples, we are talking about, relative abundance, because that PCR is biased towards certain taxa, right? Some microbes want, the regions of that we are looking into won't be amplified as easy as for the other microbes, right? So then we are looking at that relative abundance, and we don't understand a full complexity of that sample. We only understand what, the identification. We only identifying the species that are out there. That is, if we can identify to the species level, right? Sometimes we can't even identify to the species level because we are limited, to the size of the target, and maybe that target that we have is not differential enough, right? But with NGS, we can do more than one target at the same time. So if you don't want to do shotgun metagenomics approach where it's too much data, too much information for you, but you still want to look into what bacteria and what fungi are in the same sample, you could do those two different regions, even possibly in one sequencing run. We just have to think how to do it, but it's possible, right? When it comes again to targeted sequencing, we can look into something very, very specific like TB antimicrobial resistance, right? We can do this with whole genome sequencing, right? Having sequencing a whole genome TB, but we can also have it. We can also do this in a targeted approach, right? Where we're amplifying the antimicrobial resistance genes, right? We can amplify those, create a library, sequence that, and then check which microbes are resistant to one antibiotic, and this obviously will help us to treat the patient faster if we know that specific organism that person is having is actually resistant to the drug that the doctor is prescribing. They can prescribe a different one, and obviously this makes it much more time efficient when it comes to the disease control and of course cost efficient as well, right? And we can do similarly with HIV genotyping, right? Where we are looking into integrase inhibitors and which we're looking into drug resistance for this and which strains are resistant, right? Same principle applies. We want to do it to treat a patient faster and more efficiently. When it comes to viral sequencing, like I mentioned briefly before, we can obviously work with RNA viruses. In that case, we need to convert them to cDNA. And if we're going to work with Illumina DNA prep, that DNA needs to be double-stranded DNA. We would prepare a library, sequence it, data analyze it. If we are working directly from let's say RNA virus, we can also employ a different kind of library prep that is all-inclusive. So through-seq RNA library prep is all-inclusive. It actually has that cDNA synthesis portion in it. Again, it's all just to prepare a library so that we can sequence it. Or if we are working with DNA viruses, either single-stranded or double-stranded, again, we can employ Illumina DNA prep. So in essence, what I'm trying to say is that, if we're working with viral sample, either being DNA or RNA, we must convert it to cDNA first, or we can amplify that virus out, either with very short overlapping fragments or having a long-range PCR where we amplify the whole region at once. We prepare a library and sequence it, right? And this is what we employed when it comes to COVID-seq, right? Everybody is talking about SARS-CoV-2, and it's the most famous virus in the world right now. So it's only natural that Illumina has a test when it comes to diagnosis for it, but also you will see just now they came up with the surveillance solutions as well. So when we talk about COVID-seq, we are amplifying a whole genome of SARS-CoV-2, and we do that in 98 amplicons. So we have 98 amplicons that are overlapping, and that will in the end give us bioinformatically later on, we will patch it up and it will give us a whole genome of SARS-CoV-2. And obviously, when it comes to specifically to diagnostic tests, this was developed specifically for larger instruments like NovaSeq and NextSeq 550, NextSeq 2000, where we could do anything from 384 samples to 3,000. More than 3,000 samples, right? But not every lab out there has that kind of capability, right? So Illumina quite recently, actually a month ago, less than a month ago, came up with a smaller pack, right? Previously we had a very large assay pack. Now we have a 96 kit size, right? And it's actually the same kit. We're still dealing with the amplicon based, where we are amplifying our SARS-CoV-2, and this kit actually now includes the indexes as well. So it's a full solution, right? When we're talking about it, you just obviously need to have your samples ready, right? You need to collect the samples, and then you have to have RNA extraction done. But that COVID-seq actually starts with cDNA synthesis. So it's all inclusive from cDNA synthesis point of view. You would have cDNA synthesis, then we would have two pools of these 98 primers where we would amplify a whole SARS-CoV-2. We would then proceed to further library prep steps, like adding the adapters onto those amplicons so that we can sequence it actually on any Illumina sequencing instrument. If you have iSeq, MySeq, MiniSeq, NextSeq, 550, 2000, NovaSeq, any of the libraries that you prep for Illumina can be sequenced on this instrument. Obviously, depending on the capacity of the instrument, right? Something like iSeq, maybe we can only do 4 SARS-CoV-2 samples, but for example, with a MiniSeq or a MySeq, we could do up to 25 different samples. When we're talking about higher capacity instruments, like I mentioned before, we can push that to up to 384 samples on the NextSeq 550, for example. Illumina has, in the base space, they have a COVID lineage app, and I'm not going to go into too much detail into this because we will touch on this on the 27th of August when we have a bioinformatics session. But just to show you that this is actually open to everybody, right? There's no charge for this specific app in the base space, but with this pipeline, we are able to annotate mutations, assign phylogeny, and assign lineage to different SARS-CoV-2 variants out there. When it comes to the last method that I want to talk about, when it comes to working with infectious diseases, I want to talk about enrichment. I briefly said that in that workflow, we will create a whole genome library, and then we will have a probes. So we'll have oligos that are specific to the target that we want to capture, right? So if you look in this diagram, you will see that our region of interest is this little purple target. We will create the probes that will bind, that will hybridize to the specific region. And because those probes have biotin on it, we will introduce strep avidin beads, right? That will bind to the biotin. And then when we have a magnetic stand, we will pull with a magnet, we will put strep avidin bead, which is bound to the biotin, which is bound to our probe, which is bound to our target of interest, right? And so we pull what we are interested in, we keep that, and we wash everything else away, right? With the amplicons, we are amplifying what we are interested in. Here, we are physically fishing it out, right? To be a little bit blunt, we are physically fishing out what we are interested in. We're washing everything else away, and we're sequencing only a subset of what we want to sequence, right? And this is ideal when it comes to, let's say, with working with co-infections, to continue with this SARS-CoV-2 and different respiratory pathogens out there. We have what we call a respiratory viral oligo panel, where we are looking at detection of very large pathogens, co-infections, right? Specifically, this kit is we are looking at 40 different viruses out there when it comes to respiratory viruses. So not just SARS-CoV-2, obviously, the most popular one, but we are also looking at influenza, para-influenza, adenoviruses, Boca viruses, rubella, mumps. So over 40 many different viruses out there. And we are doing this like I mentioned, having probes that are specific to these viruses. We are obviously, if the sample has that specific virus, it will bind to it. You will pull it out, and obviously you will sequence it. And this allows us to do it very cost effective, to look at many different viruses at the same time, are very cost effective, right? Because you're targeting certain regions, so we are decreasing the amount of, when you think about shotgun metagenomics, if you were doing it that way, you would have to sequence everything at once, right? But here we're just kind of zooming in specifically what we want to sequence. And like I mentioned to you, we will obviously create a library, right, with the Illumina DNA prep. It's part of this whole enrichment process, and then we will have the probes that are specific to this, let's say 40 different respiratory viruses that we have in a panel. We will capture those, and obviously we will sequence those, and we will analyze it. We also have something called respiratory pathogen ID panel, where we are looking at 280 different pathogens, not just those 40 viruses, but we're also looking at the bacteria, respiratory bacteria, but also fungi. So with bacteria, obviously it will be TB, but also Legionella and Listeria and many different ones. And with the fungi, we would look into Aspergillus, Fusarium, Rhizopus species. So if you guys are interested into what exactly, what pathogens are part of this kit, obviously we can share that list with you. But also what is very nice about this specific kit is that we are also looking into antimicrobial resistant genes out there, right? So we are looking into over 16 different antibiotic classes that we can identify, right? And we do this at the same time, right? So it's enormous amount of targets that we are looking into at the same time, right? And this is ideal to look, for those, to understand that co-infections, but also to understand the transmission and to look at the epidemiology studies. So just briefly to show you what is possible with this different, either just viral panel or with respiratory pathogens, where we're talking about bacteria, fungi, and viruses. It's ideal for those benchtop instruments. If you have a very small instruments like a mini-seq or a MiSeq, you can do up to 25 different samples. But if you have NextSeq 550 or NextSeq 2000, you could do up to 384 different pathogens. So it's ideal to do that, to track transmissions and co-infections. And just to summarize on the COVID-seq portfolio that Illumina has, we spoke about 4 different approaches out there. Obviously, depending on which ones you utilize, you would be able to just identify and characterize SARS-CoV-2 or the different respiratory pathogens in there, but others you would be able to detect and to monitor evolution, and all of them are actually able to do the screening for you of just SARS-CoV-2 or other pathogens. And just to summarize about all the techniques that we were talking about and all the applications we were talking about, when it comes to infectious diseases, but not just infectious diseases, not just human infectious diseases, but also animal infectious diseases, but also for those of you that are working with environmental samples, or maybe agriculturally important species, food production, and so on. It's really important that you choose the correct technique that is right for your biological question, right? And you will notice here, if we have a targeted approach, right, that we might not get up to the species level if we are working just with a small fragment, if we are looking into just a very limited amount of data, right? We might identify it up to a genus level, but if it's crucial for us to identify that species up to a species level, then obviously we need to look into either larger region, or we need to look into multi-locus sequence typing, where we're looking at many different regions at the same time, and this allows us to identify it, right? But if we are interested in variant detection, strain detection, clone detection, then obviously we need to look into either enrichment approach, where we are physically pulling out those specific targets where we are interested in, or ideally we are looking into whole genome sequencing, and we are sequencing everything that is in that sample. And obviously you will notice that as deeper we go into the, let's call it identification, the specificity, the nitty-gritty of the organism, obviously more data we need, right? If we just want to understand what is the group of organisms that we are dealing with, we don't need a lot of data. But if we need to assess, understand those microbes, look at, monitor viral changes, bacterial changes, monitor, look into that evolution and rearrangement. And obviously we need to zoom in more and look at the whole genome and understand it on the whole genome level. I want to thank you, everybody. I know we can talk much more about infectious diseases in NGS, but I think for today we'll call it. If anybody has any questions, please feel free to either put it in a chat or unmute yourself and just ask. If there is a need, we can also set up one-on-one meetings. If you guys want to discuss your specific NGS projects, please reach out to us. Any email that ends with @separations.co.za, we will point you to the correct person to which you can discuss your project. So please feel free to reach out to us, and don't be shy when it comes to NGS. Colette, are there any questions?
None that I can see in the chat at the moment.
There was just one person who asked where they can access the previous seminars, so we can maybe mail her the links. Or I'm not sure if everyone wants it, we can maybe post it in the group chat. But yeah, I'm not sure.
Yeah.
What do you think?
Yeah, they're available on our YouTube channel. Yes. Yes, and actually it would be good for those guys that are new to NGS to maybe start with those ones, because like I mentioned, we are continuing from where we left off last year, and we're going a little bit more into nitty-gritty stuff, not so much basics. But yeah, we can definitely share those with anybody interested.
Any other questions, ladies and gentlemen? You've got the specialist at the moment. Then if there's no questions, Marija, thank you very much for the presentation and everything you shared with us. And then you will receive an email with next week's session for same time, same place. And that will also be presented by Marija next week. So thank you very much for your time, and we'll see you next week.
Thank you, everybody. Have a good